Well, I think the major difference is that the organs that we typically think about, like the heart and the lung and the brain and the kidney, are well circumscribed so that they're physically and geometrically intact and definable with the naked eye, sometimes with a microscope, but typically with the naked eye. Whereas the endothelium is systemically distributed throughout the body, right from the head to the toes, and so because it's so disseminated and because it's so thin, it's an extremely thin layer. It's really difficult to observe. Certainly with the naked eye, and even under microscopy, it's sometimes difficult to appreciate that it's a continuous, integrated system. So that, I guess, would be one difference. The second difference is that most organ systems comprise more than one cell type, so that the heart has cardiomyocytes and endothelial cells and fibroblasts and macrophages and other cell types within the ventricular and atrial walls. The liver has hepatocytes and endothelial cells and macrophages called Kupffer cells and other cell types. So the endothelium, if one were to look at it as an organ system, is unique in the sense that it consists only of one cell type. The endothelium has many different functions. One of them is to control permeability or transfer of substances, nutrients, electrolytes, gases, growth factors, and other substances, gases from the blood to the underlying tissue. So control of transfer of substances from blood to tissue and from tissue to blood. It also controls what we call the hemostatic balance, which is the fluidity of the blood. It maintains blood in a fluid state so it continues to flow, and it prevents excessive blood loss when there's a rupture in the integrity of the vascular wall. It's involved in trafficking of white blood cells, which circulate in the blood but which are required in tissues to fight infections. It's involved in new blood vessel formation, what we call angiogenesis and vasculogenesis. It's absolutely critical to innate and acquired immunity, so endothelial cells are able to respond to inflammatory signals and to bacteria, to viruses, to fungi in ways that are adaptive to the host. Endothelial cells also are critical during embryonic development to the development of certain organ systems like the pancreas and the liver. And so that's just a small sampling of the many functions the endothelium carries out across the body. My feeling and what I have always emphasized is that the endothelium, in my view, is involved in every human disease state. Either as a primary determinant of pathophysiology, so it either drives the disease, or it's a kind of a victim of collateral damage in the sense that it is affected or influenced by the disease, and in turn can then amplify the pathophysiology of that disease. And the reason I say that, first of all, is the literature, which has over 300,000 articles related to endothelial cells and endothelium, provides compelling evidence for that fact, that it's involved in all diseases. And secondly, the endothelium, because it lines capillaries and all blood vessels, is no more than just a few microns away from every tissue cell in the body, and it's a highly adaptive cell that responds to its environment. So, by definition, any change that occurs in disease and pathology in a tissue is going to somehow be felt by and sensed by the local endothelium. The endothelium is really difficult to study in humans, let alone in animals. It is, because it's so systemically distributed, as are capillaries and blood vessels throughout the body, and because it's so exquisitely thin, it's not well circumscribed like the liver, like the spleen, like the gallbladder, that it's very difficult to image using conventional radiological techniques, and it's very difficult to assay in the types of ways that we assay other organ functions. So it kind of flies below the clinical radar screen. You know, clinicians and physicians are trained to think about the heart as an organ and the liver and the lung as an organ, but they're not trained to think about the endothelium and the vasculature as an organ. A second problem relates to the fact that we tend to think as physicians and scientists of the vasculature of blood vessels being comprised largely of arteries, and our focus is on the vessels that we can see with our naked eye and that are important in acute diseases that cause most of the mortality in the Western world, which are stroke and acute myocardial infarction. So our tendency is to think about large vessels, particularly arteries, which comprise a tiny fraction of the surface area of the endothelium and the mileage of the vasculature throughout the body. And I think the third reason why there's such a gap between bench and bedside in endothelial biomedicine is because the endothelium is incredibly complex. It's very adapted to its environment, so that an endothelial cell in my lung is very different from an endothelial cell in my liver, which is very different from an endothelial cell in my kidney. And that complexity scares people and paralyzes people and makes it very difficult to get one's arms around in terms of defining what an endothelial cell is and defining diagnostic and therapeutic parameters around the endothelium, because every endothelial cell is different. So I think for those three reasons, it flies below the clinical radar screen. We think largely about the arteries and veins and ignore the capillaries, and the third being that it's very adaptive, kind of like a chameleon, that marches to the tune of the local environment, just makes it an organ that's less familiar to most people. Well, I think it's to bridge that bench to bedside gap, there has to be a better way of developing in vitro-based assays that we can do outside the body of the animal or the human, where we can recapitulate the environment in such a way that the endothelium is behaving close to what it is behaving like in the in vivo or natural setting. So that's one of the biggest problems. The third point that I made about the bench to bedside gap was its complexity, and part of the problem is that when you remove an endothelial cell from its native organ or its native tissue, it's like a fish out of water. It drifts. It becomes a shadow of its former self, so that its phenotype in vitro is very different than in vivo because it's lost its environmental cues. So the biggest challenge for us is to understand how to recapitulate this environment in vitro so that it parallels or mimics its native environment. Because most of endothelial biology is focused on cell culture, where we don't make that attempt to recapitulate the environment. And that's why there's such a big gap. I can publish 1,000 articles in my career on these in vitro cell cultures, but it's meaningless in terms of understanding the in vivo correlate. That's the biggest challenge we have, I think. I think tapping its therapeutic potential to its fullest is a long way off. I mean, I think we're already seeing some medications being used that have an endothelial effect. Statins would be one example. I think statins, which we use in patients with hyperlipidemia and which reduce lipid levels and are felt to be kind of have a healthy effect on the vasculature, almost certainly exert some of their effect by attenuating endothelial activation and endothelial dysfunction. So that's one class of drugs that I think has already proven to be quite beneficial, and that, to me, is one form of endothelial pharmacology. But I think that moving forward, I think that the opportunities are endless in terms of developing pharmacological agents that can target not just the endothelium as an organ across the vascular tree, but different vascular beds, being able to target endothelial cells that are misbehaving in one or another organ. Huge potential, huge potential. So endothelial biomedicine was an idea that I had a few years ago, where I felt it was necessary to promote an awareness among scientists and clinicians about the importance of the endothelium as a systemically integrated organ system. And I felt that to promote that awareness, that it was important to bring in multiple branches of science. So the uniqueness of the book comes from the fact that not only is it focused on the molecular and cell biology of the endothelial cell, but also the biology, physiology, and pathophysiology of endothelial cells from different vascular beds, so that we have components that are related to endothelial cells from the lung and endothelial cells from the brain, and you go through the different traditional organ systems, and there are detailed chapters related to each of those vascular beds. But more than that, it's a book that brought in other fields, so that there's significant sections that discuss the history of medicine, the history of endothelial biology, the history of cardiovascular medicine is one angle. A second angle is the evolution of the endothelium in the cardiovascular system. So to bring in, I brought in a lot of chapters that draw on our knowledge of comparative physiology and anatomy across species and across time. And then a third kind of unusual outside-the-box component that I brought into the book was complexity theory, and this whole idea of network theory and dynamical systems modeling, and the use of metaphors to describe the endothelium. So the effort here was kind of to jumpstart the field and to have something in that book that was palatable and interesting to many, many different fields, and have people start to talk about it and to think about the endothelium in new ways. So it was kind of unique in that, in those fashions. It was a little bit of an outside-the-box attempt to bring many, many different fields together in a way that felt thematic and integrative. I think today that our compartmentalization of the body is along the lines of very traditional organ-specific disciplines. So we have cardiologists, and we have nephrologists, and we have pulmonologists, we have neurologists. But I think what we're beginning to appreciate are patterns of communication between different organ systems that cross traditional boundary lines. And I think the vasculature is one classic example of that, where we have diseases of the vasculature that affect multiple organs and multiple vascular beds at the same time. And in the absence of an integrative discipline, except pediatrics, which does look at the body in a holistic way, but in the absence of integrated ways of looking at the human body in adults, things get missed and things fall through the cracks. And I think that that's the value of vascular and endothelial biomedicine. I think the one concern of endothelial biomedicine, so I remember approaching somebody to do a chapter on pulmonary hypertension, for example, and this individual's response is, well, I don't want to write a chapter on pulmonary hypertension viewed through the lens of the endothelium, because it's more than an endothelial disease. It involves the smooth muscle cell layer of the blood vessel. It involves the whole blood vessel. And so one argument may be that looking at the endothelium in isolation is a mistake, because the endothelium communicates with many other components of the blood vessel wall, and maybe we should be thinking about the vasculature as an organ system. And that argument has its merits. But again, my goal was really to promote, for better or for worse, an awareness of the endothelium as kind of the standalone entity that deserved recognition in its own right.